So a few questions now about the C6 stage part of glycolysis. So does the C6 stage come under the preparation or payoff part of glycolysis? So the C6 stage is part of the preparation phase because we're actually using up ATP to create different compounds which are needed for later steps of glycolysis. So what happens in a phosphorylation reaction? So in a phosphorylation reaction, we are adding phosphate onto our particular compound. For example, in the first step, we add phosphate onto the glucose to create glucose 6-phosphate. And where does the phosphate come from in these two phosphorylation reactions we see in step 1 and 3? So by breaking ATP down to ADP, we actually release one of the phosphates, and this phosphate is the one that's actually added onto the component. So this is how glucose gets to glucose 6-phosphate. The phosphate comes off ATP and is added onto the glucose. So what is an isomerase and how does it function? So an isomerase is an enzyme that will convert a compound from one isomer version of itself to another. So in this case, we saw in step 2 that the phosphoglucoisomerase converted glucose 6-phosphate to fructose 6-phosphate, which have the same atoms, but they're just arranged differently. So let's have a look now at the next part of our glycolysis reaction. So this is step 4, where we're actually now breaking down the 6-carbon compound, fructose 1,6-bisphosphate, and we're separating it into the two other parts, which are the dihydroxyacetone phosphate and the glyceraldehyde 3-phosphate. So in step 5, we actually convert the dihydroxyacetone phosphate into a glyceraldehyde phosphate. So now we're left with 2 glyceraldehyde 3-phosphates, which we can proceed through the payoff phase together with. So these 5 reactions together that we see here are what we call the prep phase of glycolysis. And they're the prep phase because we're using ATP up, and we're also creating the glyceraldehyde 3-phosphates, which are needed to continue glycolysis. So in the energy generating stage of glycolysis, which is coming up next, there's actually 4 ATPs made. So this is in what we call the payoff phase. So it's the last 5 reactions being the payoff phase. So specifically it's steps 10 and 7 which are giving us the 4 ATPs. So each provides 2 ATP where we're cleaving a phosphate off the component and then putting it back onto ADP, which is allowing us to reform ATP. So the reaction we're doing is ADP plus a phosphate, which is coming off the component within the reaction, and that is giving us the ATP. Overall, in our payoff phase, there's 4 ATP being produced and 2 are used in the prep phase. So this gives us an overall gain of 2 ATP every time we do the glycolysis reactions, which, remember, consist of these 10 steps. Alright, so step 4 is actually where we're cleaving up our 6-carbon compound into 2 3-carbon compounds. So we start with something that has 6 carbons and we split it into 2. So one of these 3-carbon compounds is our dihydroxyacetone phosphate and the other is our glyceraldehyde 3-phosphate. So you can see what's happened here. Basically, we've split the fructose 1,6-bisphosphate in half. So half of our component here in the pinky color is dihydroxyacetone phosphate. So where it was attached to carbon, it's now accepted a hydrogen in place of this. Whereas what's in green is actually the glyceraldehyde 3-phosphate. So to accommodate this, what's actually happened is the hydrogen that was associated with this oxygen, so it was an OH at the end here, and hydrogen like this, so we've actually lost that hydrogen, and that's the hydrogen that's been given to the dihydroxyacetone phosphate. And to satisfy the bonding arrangements within glyceraldehyde 3-phosphate, the oxygen now forms a double bond to the carbon instead. So you'll notice that each of these components actually contains a phosphate because the phosphates were attached at either end of our fructose 1,6 bisphosphate. So that's why they both have a phosphate in their name, because they both have a phosphate attached within those 3 carbon compounds. And we have this reaction being facilitated by something called an aldolase. So an aldose is a sugar that contains the aldehyde functional group. So we can see that we have the aldehyde functional group present in our glyceraldehyde 3-phosphate up here. So that's how it's getting its name here. It's creating this aldose, which is our glyceraldehyde 3-phosphate. By comparison, the dihydroxyacetone phosphate is actually a ketone, but the name of the enzyme actually came from the formation of this aldehyde. Alright, so step 5 is to try and convert our dihydroxyacetone phosphate into a glyceraldehyde 3-phosphate, because remember we need 2 glyceraldehyde 3-phosphates so we can enter the payoff phase. So all of the atoms that we require are already there, so all we need to do is another one of these isomerization reactions. So to do this, we do a triose phosphate isomerase, where we're basically just moving the position of the carbonyl group, which is our C double bond to O. So here's the carbonyl group, so we're moving it from the middle to the end. So this rearrangement is actually turning the dihydroxyacetone phosphate from a ketone into an aldehyde. So we're actually altering the functional group by rearranging these atoms through the isomerase. So remember, an isomerase is an enzyme that is able to change the isomer form of the compound. So in this way, we're actually changing from structural isomer number 1 over here, and over here we've got our structural isomer number 2. Alright, so now that we've got our 2 glyceraldehyde phosphates, every single reaction we're going to do is we're going to be doing it twice, but we're only going to show it once. But just to understand, from 6 to 10 in these steps, we don't just do the reaction once, we do it twice, because there's 2 of these 3 carbon compounds. So just going back to where we had the summary of the whole thing. So we can see at the top here, all these reactions we've talked about so far, these all contain, the top 3 contain our 6 carbon stages, and then we did another couple of steps to create these 2 glyceraldehyde 3-phosphates. So now we're going to be doing the same reactions with these 2 glyceraldehyde 3-phosphates through steps 6 to 10. So although we are only showing the reaction once, we're going to do this reaction 2 times for every time we're going through the glycolysis reaction set. Alright, so the first thing we need to do is actually an oxidation reaction. So it's going to use our NAD+. So what function does NAD+ serve? So NAD+ facilitates our redox reaction. So depending on the form of our coenzyme here, it's actually either going to be facilitating an oxidation or reduction. So remember, if it's in the form of NAD+, we say that this is electron poor and also hydrogen poor. So we need to change the NAD+ to a form where it actually has more electrons and more hydrogen. So we call this the NADH, and this is the electron and hydrogen rich form. So to do this, we actually have to take electrons and hydrogen off our component within the glycolysis reaction, and we're going to give that onto the NAD+. So that's what we're doing here. We're oxidizing glyceraldehyde 3-phosphate through using NAD. So remember, in a redox reaction, one component is oxidized and one is reduced. So in this case, the NAD is being reduced because it is accepting hydrogen and electrons, whereas we have the glyceraldehyde 3-phosphate being oxidized because it is losing hydrogen and electrons. So we can see where the glyceraldehyde 3-phosphate loses the hydrogen and electron. So it's actually in this location here where I put the box around. So this is actually where we're able to attach the phosphate, and we actually use this phosphate connection here. So the phosphate is actually coming from an inorganic source, so we don't actually need ATP to add the phosphate on in this case. The reaction is being driven by the redox potential of the coenzyme here, which is NAD+. So the enzyme that actually facilitates this reaction is called glyceraldehyde 3-phosphate dehydrogenase. So the dehydrogenase part of the name basically tells us that we are removing hydrogen. So what are we removing hydrogen from? We're removing hydrogen from the glyceraldehyde 3-phosphate, and that's what we see in the first part of the name. So once we've removed the hydrogen from the glyceraldehyde 3-phosphate, it gets passed on to the NAD, which makes it NADH, and now we have this free position within our glyceraldehyde 3-phosphate, which can bond to the phosphate. And by doing this, we've actually now got two of these phosphate groups attached. So in later steps, we're actually going to start chopping these phosphates off and recreating ATP. So this is how we generate ATP in the payoff phase. So the next step is the first one of these reactions where we're creating ATP. And we do this through what we call a substrate level phosphorylation step. So to do substrate level phosphorylation, we actually need an ADP, because this ADP is what we're going to add our phosphate onto. So we need the ADP to add our phosphate onto. It's going to accept the phosphate and become ATP. So we cleave off one of the phosphates from the bisphosphoglycerate, and that actually occurs at the carbon 1 position. So the one that is shown here in blue. So it's actually the phosphoglycerokinase that will actually clip this phosphate off, and it will pass it on to ADP. So by adding our phosphate with the ADP, we're actually able to create an ATP. And we can see what's left over. We now just have the one phosphate attached, so it's not bisphosphate anymore, it's just phosphate. Remember, bis means 2, but we've removed one of the phosphates here, so we only have 1. So here we have 3-phosphoglycerate. Alright, so to summarize what happens in substrate level phosphorylation, we cleave off the phosphate from the compound, in this case it's 1,3-bisphosphate glycerate, and we give this phosphate onto the ADP to make ATP. So how many times do we perform this reaction? Do we perform this reaction once or twice? So how many 1,3-bisphosphate glycerates do we have? Remember, we're actually in the C3 stage of glycolysis now, so that means we have 2 of these 3-carbon compounds, 2 1,3-bisphosphate glycerates. So we actually have to do this reaction twice. So because we do this reaction twice, we're actually able to give another ADP, a phosphate, which means we're again creating ATP. So we count these up, 1 here and 1 here. So overall, we're creating 2 ATP by doing the step 7 of glycolysis twice. So the next reaction is another isomerase reaction. So basically what we're doing is we're moving the phosphate from the C3 carbon, which is here, to the C2 carbon, which is here. So we're just switching what these carbons are attached to. So on the left-hand side, the carbon is attached to an OH, but on the right-hand side, the carbon is actually attached to an oxygen and then to a phosphate. So you can see that the carbons 2 and 3 have basically just swapped what they have attached to that carbon. So when we're 3-phosphoglycerate, the phosphate is actually attached to carbon 3. But when we've changed this to 2-phosphoglycerate, we've actually moved the phosphate onto carbon 2. So we call this a phosphoglucomutase enzyme because we're basically just changing the position of the phosphate. So this is another isomerization reaction. So we're changing from the structural isomer in the first form, which we see over here. So it's structural isomer number 1, and then we're creating the structural isomer number 2. So that's why we call this an isomerization reaction. So is there any need to add any other atoms or remove any atoms when we do an isomerization reaction? No, there's no need, because all of the atoms that are already required to make the product are already in the reactant. All we have to do is actually move these atoms around to form the new product. So that's what we've done here. We've moved the phosphate onto carbon 2 to create our product. So step 9 is actually a dehydration reaction. So we saw dehydration reactions when we were talking about our functional group. So what actually happens when we dehydrate something? You can probably relate this to when you're feeling dehydrated. It's basically just the removal of H2O. So we remove the atoms that make up H2O. So which are the 2 groups of atoms that make up our H2O? It's our OH and also the hydrogen. So these 2 together will form H2O. So what we're actually doing is removing the pink atoms here to make our water. So by doing this, we actually change the compound from an alcohol into an alkene, because when we actually remove that hydrogen and OH from these carbons, that actually means they need to form new bonds. So there's new bonding positions available for these 2 carbons. So instead of forming new bonds with other atoms, what they actually do is just form an extra bond to themselves, which is what we see here in blue. So in this way, we've changed the alcohol into the alkene. And the byproduct of this reaction is water. So by doing this, we've actually converted the 2-phosphoglycerate to our phosphoenol pyruvate, which is our second last product of our glycolysis. So we can actually see it actually contains pyruvate in the name there. So we're getting pretty close to the end. And the enzyme that we use to do this process is actually called an enolase, and it requires a cofactor of a magnesium 2+ ion.